Good evening, everybody. In today's lecture, we learn about myasthenia gravis. What is myasthenia gravis? The myasthenia stands for weakness and gravis stands for serious. The myasthenia gravis is a chronic autoimmune disorder, which involves the serious weakness of the voluntary muscles. The myasthenia gravis is a disease related to the neuromuscular junction. Now, what is a neuromuscular junction? So, to understand this disease, we need to understand what is neuromuscular junction, its normal structure and function. A neuromuscular junction is when the motor nerve ending makes a connection with the muscle fibers of the voluntary muscle. For example, this is the motor nerve ending. Okay. And here, this is the muscle fiber of the voluntary muscle, say your bicep. Okay. So this is the presynaptic component. This is the synapse. And this is the postsynaptic component. So the presynaptic component is the motor nerve ending, here it is synapse, and the postsynaptic component is the muscle fiber of the voluntary muscle, say for example the bicep. And in the presynaptic nerve ending, we have vesicles. We have certain vesicles which stores the neurotransmitter called acetylcholine. We can say that the motor nerve endings synthesizes and stores the acetylcholine. Now, how it synthesizes the acetylcholine? First of all, the choline is obtained from a diet. It is absorbed into the GI tract, it comes into the circulation and then into the extracellular fluid, and it is taken up by the motor nerve endings through a transporter called choline transporter. Through this transporter, the choline enters into the presynaptic component. Also, in the presynaptic component, we have mitochondria. This mitochondria releases acetate or the acetyl coenzyme A. So this choline fuses with the acetyl coenzyme A with the help of the enzyme known as choline acetyl transferase, and it is stored in the vesicles. Now, when the action potential occurs, that is when the wave of a depolarization is followed by the wave of a repolarization. Depolarization means when the positive ions, that is sodium and calcium ions, enters the presynaptic nerve ending. So when the depolarization occurs, there is the influx of the sodium ions. So this influx of the sodium ion activates depolarization sensitive calcium channels. Now, what happens that there are special proteins on the membrane of the vesicles which stores the acetylcholine. Here, there are special proteins on the vessels which store the acetylcholine. These proteins are called as synaptobrevin. This protein is called as synaptobrevin. And also there are special protein located on the membrane of the motor nerve endings. The ending of or the membrane of the motor nerve ending is known as, this membrane is known as neurolemma. So we can say that there is a special proteins located on the neurolemma which is known as syntexin. Okay. We'll draw the structure of syntexin over here. This is a syntexin. It has two main domains here. correct? It has two main domains. So what happens that when the calcium enters the presynaptic nerve endings, these domains get loaded with the calcium. When these domains get loaded with the calcium, they start to repel each other and forms an open structure like this, like it opens its mouth. So when this occur, immediately the synaptobravin, which is a protein located on the vessel of the acetylcholine, fuses and locks with the syntexin, and it undergoes the conformational changes. So during this, the membrane of the vessel fuses with the neurolemma. Uses with the neurolemma. When this fusion occurs, a small hole is generated on the membrane through which the acetylcholine is released in the synapse. Now the concentration of acetylcholine is much higher in the upper side, so it diffuses from a higher concentration to lower concentration. And it approaches the postsynaptic membrane, which is the folds of the muscle. These folds of the muscle are known as a sarcolemma. These folds of the muscle are known as sarcolemma, and it has crest. These are known as crest. The uplifted portion are known as crest, and these crests are loaded with the cholinergic receptors. These receptors are ion channels. Okay, these receptors are the ion channels and a pentamer. Pentamer means they have 5 subunits. They have 5 subunits and they have 2 pockets. So when this acetylcholine binds on these 2 pockets, this central pore gets open and the cation enters the channel, which is sodium. When the cation and the sodium enters the postsynaptic membrane of the muscle, the resting membrane potential become partially neutralized. Why? Because the resting membrane potential is normally negative. Say it's a minus 90 millivolts. So when the sodium enters into the postsynaptic membrane, it may become minus 50 millivolt or minus 40 millivolt. So this small fluctuation in the resting membrane potential is known as the end plate potential. When this end plate potential approaches a threshold, okay, so it activates another voltage sensitive sodium channel. Now another voltage sensitive sodium channel. So this is also a sodium channel and this is also a sodium channel, but this is the acetylcholine sensitive sodium channel and this is a voltage sensitive sodium channel. So what happens, again there is a heavy influx of the sodium inside the postsynaptic membrane, and when the positive ion enters the membrane, it undergoes the wave of depolarization, and this wave of depolarization spreads through the sarcoma. It goes into the T tubules, and calcium is released into the cytosol, and it leads to the contraction of the muscle, and the actin and the myosin sliding. So here we can see that the electrical energy is being converted to mechanical energy. Okay. Now once the acetylcholine action is completed over here, so it is taken up by an enzyme which is known as acetylcholine esterase. These enzymes digest the acetylcholine after the completion of the function. Now this entire process is known as neuromuscular junction transmission. Okay. So the diseases that impair the neuromuscular transmission. Okay. So one of the hallmark findings finding or the clinical feature of the diseases that impair the neuromuscular transmission is that it produces a weakness of the muscle, but which is painless. It is painless weakness. The patient complains of the weakness, but it is painless. So, which diseases cause the impairment of the neuromuscular transmission? So, first is the autoimmune. So, in the autoimmune, we have the myasthenia gravis. So, in the myasthenia gravis, 85% of the patient have the antibodies against the acetylcholine receptors. Against the acetylcholine receptors over here. They have the antibodies against the acetylcholine receptors, and the remaining 15% of the patient has the antibodies against a special protein which is known as the musk protein. The musk stands for muscle specific tyrosine kinase receptor. So, in a 15% of a patient, the antibodies are produced against the musk proteins. Now, what is the major function? Okay, so first we'll see that where the musk protein is located. So, musk protein is also located in the postsynaptic membrane. One of the major function of the musk protein that it controls the density of the acetylcholine receptor in the postsynaptic membrane. And if the antibodies are formed against this protein, then what happens? The density of the choline nergic receptor on the postsynaptic membrane is altered, and it reduces in number. So how this happened, we'll see this in detail further. It will have a quick overview about the Lambert-Eaton syndrome. About the Lambert-Eaton syndrome. Okay, so in Lambert-Eaton syndrome now, it is also an autoimmune disorder, but the antibodies are not formed against the acetylcholine receptor. The antibodies are not formed against the musk protein. The antibodies are formed in against the presynaptic component, which is the calcium channels. The antibodies are formed against the calcium channel. So what happens? There is no influx of the calcium, and it leads to the impairment of the neuromuscular transmission. Then we will see about the congenital myasthenia gravis. Now, congenital myasthenia gravis is very rare, and it is inherited, in which the inherited defective gene makes the defective protein. Which proteins? The proteins of the presynaptic component, say the calcium channels are defective, or the proteins of the synapse are defective, or the proteins of the postsynaptic membrane are defective, say the cholinergic receptors. So what happens if all this result in an impairment of the neurotransmission? Then we have toxin mediated. So in the toxin mediated, we have two toxin, that is Clostridium botulinum and the curare. Now the Clostridium botulinum is a gram positive anaerobic bacteria that produces botulinum toxin. Now, this botulinum, Clostridium botulinum, is mainly found in the home cooked canned foods, and the botulinum toxin is a protease enzyme which acts as a scissor. Where it acts, this protease enzyme will digest this synaptobravin. This protease enzyme will digest the synaptobravin. So as a result, the membrane of the vesicle won't be able to fuse with the neural image. There will be no release of acetylcholine in the synapse, and the neurotransmission will be impaired. Okay. Then we have the curare. A curare is a plant toxin. So it was earlier used for the hunting purpose, means the fishes used to throw them into the water bodies, and the fishes used to get paralyzed, come on the surface, and the fishes used to catch the fish. Then also it was used in the arrow hand. Means the animals not only used to get injured because of the arrow, but also used to get paralyzed. Then of course it was used for the war purpose. So what is the main mechanism of action of curare? That is blocks the acetylcholine receptors, and it inhibits the binding of the acetylcholine with the receptors. Then we have certain drugs that may impair the neuromuscular transmission, although it is very rare. If it would have been frequent, these would be defined as a toxin, but this is very rare. So these drugs are aminoglycosides, quinine, procainamide, beta blockers, and tetracycline. So it becomes obvious that the patient suffering with the myasthenia gravis should avoid this medication, that is aminoglycoside, quinine, procainamide, and beta blockers, and tetracycline. So now we'll study in detail about myasthenia gravis. Myasthenia gravis. Although myasthenia gravis can occur at any age, but it has a bimodal age distribution. Bimodal age distribution means bimodal age distribution means the incidence of causing the myasthenia gravis is at the peak during the younger adulthood, that is between the 20 to 30 years, and the prevalence or the incidence of causing the myasthenia gravis is at the peak in the later adulthood, say 40 to 50 years and onwards. Okay. So when the prevalence of the myasthenia gravis is higher in the younger adulthood, the females are more prevalent, and if it occurs in a younger adulthood, the males are more prevalent. Okay. And these females with the myasthenia gravis have the thymus abnormality called thymic hyperplasia. Thymic hyperplasia. And if it occurs in a later adulthood, then male are more prevalent, and they also have the thymic abnormality, which is known as a thymoma. Thymoma. Okay. Now, what is thymoma and what is thymic hyperplasia? We'll discuss in detail further. First, we'll see about the mechanism of action in detail of myasthenia. So, as discussed, it is an autoimmune disorder. And this is the presynaptic component, which is the motor nerve ending. This is the sarcolemma, and these are the crest. And on the crest, we have the cholinergic receptors. Since it is an autoimmune disorder, it is caused by the antibody IgG. IgG. Now the IgG can pass through the placenta. So if the mother is suffering from the myasthenia gravis, this antibody can pass through the placenta, and even the newborn will have the muscle weakness. So however, these IgG antibodies are divalent in nature. What do you mean by divalent? That the one antibody can bind to the two antigens. Now, antigens in a case of myasthenia gravis is our own receptors. Okay, so this antibody will bind to one antibody binds to two receptor. One antibody binds to two receptor. So this leads to the clustering of the receptors. Clustering of the receptor, or cross linking of the receptor. It leads to the clustering of the receptor or the cross linking of the receptor. Now what happens that the cells are very smart. They are very clever. Whenever they see that there is an abnormal protein complex, what they do, they try to internalize the complex. Now what do I mean by internalization? So there are special proteins inside the membrane. They bind here in the end side of the receptor, and they try to pull the entire complex inside. Why? Because the membrane is having abnormality on its surface and it want to heal itself. So it will catch, arrest, and pull itself inside, just like the endocytosis. Okay. Like for example, these are the receptor and this is the antibody. It will get pinched off inside, and it will become a vesicle. So inside the postsynaptic membrane, we have the lysosomes. So these lysosomes will fuse with this membrane of the vesicle, and it will release the enzyme that will catabolize and destroy the antibodies. Now this mechanism occurs in a healthy individual. Now what is wrong with a myasthenia gravis patient? This clustering and the cross-linking is accelerated, which result in the accelerated endocytosis and accelerated destruction. The destruction is so fast that the membrane is not able to compensate and maintain the high concentration of the cholinergic receptor on the postsynaptic membrane. So, as a result of which, the postsynaptic membrane becomes a deficient in the acetylcholine receptors. So how this antibodies bind with this receptor, we'll see in detailed structure. Okay, like for example, this is one crest of the sarcolemma. It is having cholinergic receptor, right? And it is having an antibody. Okay, so when this antibody combines with the antigen, its tail becomes active. Its tail becomes active and starts waggling. When it starts waggling, it activates another complementary protein. What are complementary proteins? Complementary proteins are the special membrane bound soluble proteins which are found in a biological system and are mainly found by liver. So it activates the complementary protein and it gets attached here. C1, and we have the C4, and then we have the C2. The C4 and C2 activates the C3b. We have the C5b and up till 9. Oops. C7, C8 and C9. So this entire thing is called pathological channeling. Pathological channeling or ports. This entire thing acts as a drilling machine. It will puncture the membrane. Now, this entire process occurs in the entire sarcolemma. Over here, it will keep on puncturing, and the membrane will not be able to maintain its microenvironment. So, as a result of this, what will happen? It will undergo the structural abnormality. It will undergo the structural abnormality and the morphological changes. So as a result of this, what happens that the membrane or the sarcolemma becomes simplified. It becomes simplified. There are no crest. There will be no crest. And when there will be no crest, the acetylcholine receptor will not have placed to sit on the receptor. So ultimately, these acetylcholine receptors will be reduced in number. Okay. So what three things happens over here? We will again see what which three things. The first, the total surface area. The total surface area is reduced, right? The number of acetylcholine receptor per unit area is reduced, and also the gap between the presynaptic membrane and the postsynaptic membrane is increased. That is, the synaptic gap is increased. So what happens? This leads to the impairment of the neuromuscular transmission, and what it will lead to? What? Painless muscular weakness, right? So now we have seen that, we have discussed that it causes the thymic abnormality. Now, what are the thymic abnormality? That if it happens in the females, in the younger females, it causes the thymic hyperplasia, and if it happens in the adult male, it will cause the thymoma. So this is the thymus gland. The thymus gland contains the myoid cells, the T cells, the B cells, and the epithelial cells. So this thymoma is the tumor. Is a tumor. Mainly it is benign, but in some cases it may become invasive and malignant. Okay, so this is a tumor of epithelial cell. Thymoma is a tumor of the epithelial cell. And a thymic hyperplasia occurs in females, affects the B cells. Now, what is the mechanism? How our own antibody attacks our receptors? What is the mechanism? So, we know that there is a thymus. In the thymus, we have special centromal cell. Centromal cell, we have special centromal cell that are known as myoid cell. Myoid cell. Myoid cell. These myoid cell express the sarcolemmal protein on its surface. Also, they express the cholinergic receptor on its surface. In a healthy individual, what happens that when this myoid cell is exposed to the B cell and the T cell, and if these cell interacts with these proteins, they are immediately suppressed and they are killed. But in a patient with a myasthenia gravis, the thymus function is abnormal. The thymus gland function is abnormal. So what happens, instead of a suppression, it leads to proliferation and produces even more beta cell and a T cell. And leads to the production of even more B cell and the T cell. So it comes into the circulation and it gets absorbed by it acts against the neuromuscular junction, and it attacks our cholinergic receptors. So this is the mechanism in 85% of patient. In remaining 15% of patient, it is same that there is a musk protein which is present in the postsynaptic membrane. The antibodies bind to this musk protein, okay, and the again the sarcolemma becomes a deficient of the acetylcholine receptor. It will stop the binding of the acetylcholine. So this is the pathophysiology. Now, what are the signs and symptoms? Okay, the signs of symptoms of myasthenia gravis. So, it mainly involves the cranial muscles, which are the extraocular muscle. And the extraocular muscle, when it causes a problem, the patient has a manifestation of the ptosis. Ptosis means depressed eyelid. Here, as you can see, one eyelid is depressed. It can lead to the ptosis. and it can cause diplopia. Now it is diplopia that the two eyeballs don't move in the coordinated fashion. There is no coordination. There is no coordination between the eyeballs. The patient will have the double image. Okay, so it will affect the extraocular muscle. Also, it affects the bulbar muscles. The bulbar muscles are involved in articulation, phonation, and swallowing. So, if the bulbar muscles are having problem, it will cause difficulty in chewing, swallowing, and slurred speech. Okay, and okay, so myasthenia gravis. The patient with the myasthenia gravis always looks sad. They always look sad. And even if we ask the myasthenic patient to smile, they will have a sad smile. So this particular clinical presentation is known as myasthenic snarl. Myasthenic snarl. Okay. Now, some patients are lucky that in some patients there may be only extraocular abnormality, that is, they may only have the ptosis and diplopia. But some patients may have the abnormality in the neck, abnormality in the shoulder, abnormality in the respiratory tract. Okay, abnormality in the limbs, in the hip girdles. Okay, and it may even cause the paralysis of the respiratory tract. So the patient may require the mechanical ventilation, and it is known as myasthenic crisis. It is known as myasthenic crisis. So these are the signs and symptoms. Now we go to the diagnosis. The diagnosis of the myasthenia gravis is based upon the four criteria. That is, the main is the history, the patient history, that if the as discussed, that the congenital myasthenia, or if they are having any defective genes, or if any patient family members is having the history of the myasthenia. Then we have the physical examination, that is the ptosis. But during the physical examination, the patient may have the ptosis and diplopia and other physical examination, but the senses will be normal. The senses will be normal. Okay, its senses will be normal. Then we have a very famous test which is known as stencil-on test. What is stencil-on test? So it makes the use of a edrophonium. Edrophonium. It makes the use of injection edrophonium 10 mg. And when it is injected, it is edrophonium actually is a short acting choline acetylcholine esterase inhibitor. The enzyme which degrades the acetylcholine, it inhibits that. So what happens, the amount of acetylcholine on the receptor remains for a longer period of time, so the symptoms may be relieved for a shorter period of time. Now, as you can see in the image, that here the patient is undergoing tensilon test. First he is having ptosis in one eye, and when he is given edrophonium, the temporarily the symptom gets relieved. We can see the immediate improvement. So this confirms that it is a myasthenia gravis. Then we have the eyes test. Now what the eyes does, when the ice is applied on the ptosis, the ice freezes the acetylcholine esterase temporarily. And again, it blocks the degradation of the acetylcholine, and the acetylcholine remains for a larger period of time on the receptor, and the symptoms will be reversed, and the symptoms will get reduced. So these are the diagnostic tests. And we have treatment, of course. We have treatment in anticholine esterase drug, but these are longer acting. Okay, we have physostigmine, we have neostigmine. Physostigmine is given 60 mg thrice daily. It is a total dose of 180 mg. And neostigmine is also given, that is 7.5 to 15 mg starting dose every 6 hours. And as it is an autoimmune disorder. So the patient will be given with the immunosuppressants, corticosteroids like the prednisolone and the cyclosporin. Prednisolone, the starting dose may be 40 mg and up to 60 mg. Cyclosporin can also be given, that is 2.5 to 5 mg per kg. And plasmapheresis, patient can also go under plasmapheresis. Plasmapheresis means the plasma, in the plasma there are autoantibodies against the acetylcholine receptors or the musk proteins. So the during the plasmapheresis these antibodies are removed. Okay, these antibodies are removed and the patient will get relief. And the other last option is thymectomy. Thymectomy is a removal of the thymus gland. So these are the known treatment. That is, we can go with the acetylcholine esterase drugs. That is the physostigmine and the neostigmine. We can go with the immunosuppressant as it is an autoimmune disorder. We can go with corticosteroids, prednisolone, and cyclosporine. Plasmapheresis means the removal of the autoantibodies against the acetylcholine receptors and the musk protein receptor, and we can go with the thymectomy. That is the complete removal of the thymus. So that's it about myasthenia gravis. Thank you.